So I'm the last one. Thank you all for sticking around. It's been a long day. Hopefully, a lot of exciting things that you've seen, and it's kind of crazy that a lot of this started from just one student coming into my lab and me being maybe a little silly and saying that she could find a gene for her disease, but it's pretty cool what's happened. So I'm going to give you kind of an overview of where I think we're going. I'm going to start with unanswered questions. It's on the title. It was last, but it's going to be first. There's no textbook to read. There's no research article to go through to know what the right questions are. We require patients to tell us. And I think this is a really a unique opportunity for scientists. Scientists, as Courtney said, very rarely ever talk to patients. I think that's unfortunate. And it's something that certainly it's something I didn't do myself before 6 years ago. But they're an incredible treasure trove of information that can dictate how the science goes and what the questions are. Anybody who's ever come into my lab, I invite you all. Molly's over there. Her email address is griggsma@musc.edu. She just email her, not me, and she can schedule a tour of the lab. Any patient who comes in, it's a requirement. Sorry, that's the only mandatory part of people coming into my lab, is you have to write down a question on our board. We have a big glass board in the lab, and here is there's basically two halves. It all looks a little bit weird, but there's a list of these questions that the patients have. This is how we know what questions to test. And from these questions, we can actually generate hypotheses that can go into and be done at the bench. This is where we make solutions. This is where we solve problems. I think this is incredibly important. We listen to you. And that's one thing that's sort of a common theme, hopefully, that you've gained from today, is that the physicians and the scientists are hopefully listening and taking the time to listen to the patients. Because without that, you get the dismissive nature of a lot of physicians, and that doesn't work. So these are some of the unanswered questions. 3 very straightforward ones, right? What is EDS? Or what is HEDS? Genetic biomarker, diagnostics, and can we develop therapies? You just heard some very nice talks on the transauricular vagus nerve stimulation. That is one option that's really cool and really hopefully very powerful to provide to the patients. Ours is a different approach, and I think marrying these 2 things or 3 things together is going to be really powerful, and integrating with the physicians to actually make this thing happen. What is HEDS? If you read this in the articles, you kind of get lost as to what it is, because there's no real good metrics on large populations. And a lot of them are based on small case reports or small studies. We had the opportunity, because our registry is so large, we have 15,000 patients and counting in our registries. And from this, we're able to gather a lot of important information about what this disease is. So the sort of the flow chart on the left sort of tells you how we sort of parsed HEDS out from HSD patients and looking at those 2 things separately. And then on the right, you'll see sort of, this is a global registry. This is from all over the world. This is including all different races and ethnic backgrounds. So I think it's really powerful to really gain a more complete understanding of what this disease actually is. And then there's some metrics there about the ethnicity, sex, gender, and I can't read. Even though I have glasses, I can't read. Anyways, So this is just sort of giving us overall demographics. It's interesting that the vast majority of our patients are in the 26 to 44 age range. And you saw this earlier in one of the previous talks, in Dr. Wilkerson's talk. This is what we need to change. All right, for HEDS, the diagnostic timeline is 22 years. And that's on basing it on about 3,700 patients. So all these data are based on the largest registry collection of data in the world. That's just insanity. So what is HEDS? This is a 400-point question. This is developed by patients and physicians and scientists. I don't expect you to read all that. I certainly can't read it either. It provides the breakdown and the metrics of what this disease really is. It's generally categorized by physicians who are not too knowledgeable as a primary connective tissue disease characterized by stretchy skin, loose joints. Obviously, you as patients know that that's really not accurate. And so what's interesting about this registry is that really the most severe symptoms are the chronic pain, autonomic dysfunction, GI. The joint dislocations are in there, but they're further down. So shouldn't we be targeting our energy on these top things that the patients report as really the most impactful for their quality of life? I mean, how many EDS patients are really concerned with how stretchy their skin is over their GI problems, right? It's there, it's a thing, but it's not something we should really be, focused on. This to me was really eye-opening, because when you look at the constellation of comorbidities that these patients have, connective tissue, although affected for sure, it doesn't, autonomic dysfunction doesn't scream a connective tissue problem, although it could. GI could, but it's not the first thing that physicians may think of. And then triggering events. 70% of our registry reported a triggering event that after that event, they got worse. And they never recovered. This, once again, doesn't really tell me it's a connective tissue problem. It speaks more towards the immune system being a strong component. And so that's really interesting. You have puberty, so hormonal changes. You have viral, Epstein-Barr, COVID. How many patients, I've seen so many patients with Epstein-Barr, as they said that they were fine, and then they got Epstein-Barr, and they just spiraled. So what's up with that, right? Is that really a primary connective tissue problem? Could be. I don't think so, though. So we have about 20 tables of data from all of this reporting, which has been submitted for publication. So we're just waiting to hear, which is good. So then everybody can see it. I don't know if we're going to let it go out as a preprint yet, but I don't see why we shouldn't, so that the community can actually see what the data is. It also brings about the question of, is it really a subtype of EDS? And just hear me out on this. All the other subtypes of EDS are caused, except for the periodontal type, which is exceedingly rare, caused by mutations in the complement pathway. All other subtypes are truly bona fide connective tissue defects because of their genetic mutation in collagen or collagen-related genes. This HEDS, however, we have never found a genetic signal for anything related to collagen or elastin. Maybe there's a couple rare patients out there that have elastin mutations or fibronectin or collagen mutations, but we're not seeing a huge signal for that. So is it really is it really an Ehlers-Danlos syndrome, or is it something else? And I think it's, it brings about the question is it if it's not a primary connective tissue disease and it's related to something else as a cause, we need to figure that out, because maybe your body is making collagen just fine, it's just getting destroyed. And therein lies the problem. So why are you trying to fix the collagen? It's not going to help. And this might be a reason why patients have one spine surgery, second spine surgery, third spine surgery. It's just a zipper that keeps opening up. And the GI manifestations, recurrent problems. It just doesn't make sense if you think about it in that light. The triggers, the pain, GI, autonomic dysfunction, and immune cells, mast cell dysregulation, these don't scream connective tissue to me. I mean, maybe they do to some people, but maybe genetic and blood markers can provide additional clues. As Courtney mentioned, we identified this gene, it's KOK15, in 2 families, and it tracked perfectly with the phenotype in the family. It's a single missense change of KOK15. We've validated the mouse in a lot of heroic efforts. Pong is sitting back there in the back. He's a bioengineer that's did all these biomechanical testing to make sure that the things were, that the mice were a little loose, and we could, they could tie the tail in a knot. And when Courtney first handled one of the first mice, it actually turned around and bit her. So she's like, man, these things are like hypermobile. And so one of my colleagues said, oh, that's a perfect biting score. But you get yeah, all right. So that was whole exome sequencing. We know that this is going to be a minor component of the whole spectrum of EDS. It doesn't make sense based on the heterogeneity of the disease that you would have one gene that causes the disease. It just doesn't make any sense. So we know it's going to be polygenic. That means multiple different things can contribute and give rise to a phenotype. And that can vary depending on the environment and other things. So genome-wide association studies and whole genome sequencing studies are needed on a large set of patients. And so we've done that. This is not stuff I've presented anywhere else. So I thought you would like to see where we are with these sort of studies. So this is a 3 case control studies with nearly 1,800 patients. They're collected in 3 different ways. And it's complicated wording there on the screen. But what we're looking for is something in the 1,800 EDS patients that we have through the whole genome that's enriched in these EDS patients compared to our control population. It's about 6,500 control individuals that are in there that we have the DNA for. And so in the end, you get what's called a Manhattan plot. Every little dot on this is an individual SNP. We've screened 6 million individual variants across everyone's entire whole genome. This takes like a year and a half to do. It's not a trivial. And to be honest, I don't really. That's why we have a population geneticist actually doing this, because they love this stuff. It would drive me crazy. And you'll see only 2 things. So there's 2 SNPs, and we can't yet tell you what the SNPs are. I'll tell you what they do, though. SNP2, which is so the dotted line there is genome-wide significance. This is a p-value of 5 × 10 to the negative 8th. Anything above that line is deemed as genome-wide significant. So SNP2 is the most significant, but it's also the hardest to interpret its function. And so I like to sort of dumb it down for my dumb brain and go to SNP1, because that is much easier to understand. we then, since we had all this data, all this genetic data, we then, instead of looking at individual SNPs or variants or mutations throughout a person's genome, we said, well, let's just block them off as whole genes and see if if a collection of mutations within that gene are enriched in EDS. And from that, we found an additional 9 genes. And those are all represented with those arrowheads. 5 genes survived very stringent statistical analysis. And so some of those genes are really cool. But what I want you to, so GBAT stands for Gene-Based Association Test, for those who are really interested. SNP1, the one that we first identified in this GWAS, Genome-Wide Association study, it's really cool because it's involved in inflammation. It's in the brain, as well as on a variety of tissues. It is on fibroblasts. It's in the nervous system. It's in a scavenger opioid receptor. And it has so many, there's a lot known about this particular protein. And if you look at what's known about it, it's involved in pain, inflammation, connective tissues, GI dysfunction, blood pressure regulation, anxiety, depression. I mean, that sounds pretty good. So this is going to, and the other thing I think that's really important to mention about this protein is, since it is a receptor, it is druggable. So I think that there's a great opportunity to develop therapies around this. And this is something that is enriched in the 1,800 EDS patients. This is not just a single patient with a single mutation. This is something that's common among the EDS patients. So I think that that's super cool. This does not, so this once again isn't collagen. It's an immune, it's on mast cells, it's on neurons, it's on glia cells. And so that's once again saying, okay, maybe this is a neuroimmune problem, which I think that, and the other genes that we identified in that GBAT analysis are also in the same sort of pathway. So we're not finding any genetic signal, or very, very few genetic signals for collagen problems. Yeah. So genetic loci associated with pain, inflammation, and blood clotting as well, if anybody has clotting issues. Some of these are druggable, which is what we're working on now. There are some drugs out there that aren't great for this, but we're trying to improve on those. What about blood markers? So we did an unbiased, science is about being unbiased and taking an agnostic look at genes, proteins, pathways, whatever. We have no bias, right? And I will tell you that with 70% of my lab being EDS patients, they are the most critical scientists, period. Because they don't want to get it wrong. I'm like, come on, you've already repeated this like 6 times. I don't, let's move forward. We're done here. We got the answer. No, I need to make it right. Okay, well, go ahead. So what we did was we collected serum from, this was on 25, 29, 30 something patients with EDS and controls. And then we put it into a machine called a mass spec and basically let the machine say, okay, these are the proteins we identified, and then let the statistical analysis say, this is what's different between your control blood and your EDS blood. And so what came out of that was really interesting. So in the purple, I don't expect you to read this either. I'm giving you a bunch of slides you can't read. Purple and blue are 2 main, all those proteins fit in 2 pathways, the purple pathway or the blue pathway. The purple pathway is kallikrein genes, which is interesting because that's what we saw with our initial genetic discovery, was the kallikrein. Blue is the complement pathway, and Dr. Maitland might have talked about, and that is the upstream driver of inflammation and mast cell activation. And then on the right is just a heat map of individual patient level analysis. And you can hopefully see that in the EDS, you have a lot of blue boxes compared to the red boxes. And those are each individual complement proteins on the right. So C8b, C4, whatever, 8BP. Those are all individual proteins. And so this, once again, now we have genetic markers, now we have protein biomarkers in the blood that align well with each other and say that this is really more of an autoimmune, autoinflammatory, neuroimmune type of axis that we really should be focusing on. And the consequence of this is likely your connective tissue breaking down. And so that's so important, because you can sit there all day and try to make your collagen better, but if that's not the problem, if that's not the root cause, you're wasting your time if something's chewing up your collagen. And I think that's involves the sort of inflammatory pathway. We've gone on to do second and third level validation of these biomarkers. These are doing what's called ELISAs. You basically have a plate, an antibody for each one of those proteins is on the bottom of the plate. You put the blood on and then see what sticks. And then it fluoresces, and you get a quantification of how much is stuck. And so once again, we see that all the purple bars are those with EDS, and those are all the patients, and we see that once again, it's a second level validation of our mass spec findings. So the complement pathway is down, which means it's being actually all used up. It's called complement consumption. So the liver simply isn't making enough complement to keep up, and that the body is using it up at a very fast rate. Here's a really pretty graph that I'm not going to tell you about, except for J. I found J interesting. If you look at J, I don't know if you can see J. This is what might be causing problems in this pathway. And it's a response to stress, which I found really interesting, that stress can actually hyperactivate or accentuate these complement proteins. A lot of fancy things. So we're really focused on better defining EDS, what it is. Like, what is the real phenotype based on real metrics? The 2017 criteria, if anybody goes back, and I encourage you all to go back and read that paper, and you will find that there's not a single patient mentioned in that paper. There's not a single statistic. There's not a single metric yet has been adopted as a clinical tool. Our data now is on, 4,000, 5,000 patients. I think that's going to set the stage for redefining clinical criteria to redefine what this disease actually is. I think we have identified some diagnostic tools that can be used together. I don't think you're going to have one individual gene, one individual blood marker that's going to do it for you. You have to work together on these different markers. And then once again, is this a disease rooted in neuroimmune dysfunction? My bias is yes. I think there will be some patients where you can say it's probably a combination. So the last part is, how do we change the perception of the disease and impact healthcare in this population? This is, for patients out there, from what I'm hearing, is you want diagnostic tests that are molecular, that are a gene or a protein. You want physicians to be educated on what this disease is, and you want therapies. And so I've presented the first and potentially third aspect, but how do we change the perception of the disease and impact healthcare? We need to modify medical school curriculums. MUSC, 2 years ago, had one slide on EDS, and it basically stated EDS is a rare connective tissue disease. It's characterized by stretchy skin and loose joints. It's and you'll probably never see it. I obviously had a problem with that. And so I worked hard with a number of different people, including members of my lab, to really change the education of EDS in medical school curriculum. The physicians need to know, the physicians in training need to know what this disease is. It's rare. You're not going to change the perception or a physician's mind if they're, a 50-year-old physician who's been doing this their entire career. I mean, we're fortunate to have Dr. Sharma and other physicians who are knowledgeable about this and want to learn more about it, but we have to do a better job across the landscape. So we've successfully now changed education, the curriculum at MUSC and at USC as well. So across the state, we've succeeded in changing how the course is taught, how the connective tissue block is taught in medical school. Is it perfect? No. Does it need more advancement? Yes. And we will continue to work on that to make it better so EDS is recognized among physicians as they're coming out of medical school. We have the intern program. This will be my biggest legacy. I fully believe that this is the biggest impact that I can make. Besides the genetic or the proteomic discoveries and the potential therapies, training, empowering patients to change their destiny, to change the trajectory of their own lives, and then change the lives of others that they're impacting. This needs to be implemented across the country. I mean, it's an incredible opportunity. The concept of the patient scientist is brilliant. It might be my only good idea I've ever had, but it was brilliant, and it was just sort of like, we should just sort of like saying, Courtney, you want to figure out your gene? Like, we should have these patients come in and be scientists. Like, it's not a hard stretch. Why aren't people doing this? So it's, I think that's, and to see the responses from these patients, it's just, it's incredible. Lay public presentations are really important as well. Engaging Congress and NIH. Maybe I'll talk about that in one more slide. Building larger research communities. There are at least 10,000 scientists and physicians across the world studying connective tissue diseases. There's about 5 labs across the world studying HEDS. It's a big disconnect, right? How do you get more scientists doing what we're doing? I'm not doing rocket science, but I am passionate about solving this problem. We need to build larger research communities. So what we've done was we said, okay, we're going to make animal models for every subtype of EDS, just to get people engaged in EDS research and offer them for free, anywhere in the world. And so we do this through our website. So all the animal models have now been generated. So we started this about 1.5 years ago. They're all completed using CRISPR-Cas9 technology. So we've had, 500 people come through our website that register interest, and we're busy trying to follow up on some of those to get more people studying this disease. Maybe I won't have the Engage Congress thing, but every year MUSC has been gracious to highlight our successes on this disease, recognizing that it's not a rare disease, but it's truly a common disease. 1 in 500. Patel he's not here now. He got to go back to the office, but he'll say 1 in 500 FUI. He said 1 in 200 or more. Like, this is not a rare disease. People need this. EDS Society, hi, if you're listening, stop saying that this is a rare disease. It's not. We'll never figure out the actual prevalence of the disease because of being misdiagnosed, underdiagnosed, misperceived. We're never going to know what the number is. Let's just stop, cut the crap, and let's say that this is a common problem that needs to be addressed. So MUSC has been gracious to allow us to include wording in their annual white paper that they then take up to Capitol Hill to pitch to Congress through MUSC's lobbying team. And it has been for the last 2 years gotten into the Health and Human, whatever that book is called, Health and Human Labor, to engage, to request Congress appropriate funds for EDS research across the country. I can't be the only lab doing this. We need it to be funded at a national scale, and I think that will happen. It still might take a few years for various reasons. Yeah, get that joke? All right. This all started with the patients. It took me, I didn't do anything. I just, I opened my lab and my mind, and the patients are the true experts on this. And it's their unique perspectives and research insights that really drive the needle, move the needle forward. Oh yeah, there's that call to advocate. There's that long book. Anyways, so just in summary here, before we came around, before the one with a new child came around, before Courtney came into the lab, disease misconceptions, I will say that that's still evident. Little to no education of healthcare professionals, difficult to find care, lack of research, lack of awareness. These are all still things that we need to work on. But we're working on better defining HEDS. We're developing improved diagnostics so that what happens in the documentary doesn't happen again. Research and genetic discoveries, and then of course therapeutic discoveries. And then MUSC has really invested very significantly and strongly with our EDS center, which Dr. Ann Maitland is the medical director of. Mara Callanan, who's here, was a huge driver of this and a huge supporter. The CEO of MUSC, Pat Cawley, the first time that Patel and I went into his office, we said, all right, we're going to pitch this idea of doing an EDS institute. And we sat down and we had this whole slide deck prepared, and Patel said, all right, Pat, we need an EDS institute. He said, okay. We're like, wait. He's like, you just got to figure out how to do it. And so, that openness was incredible. The receptiveness, it wasn't, man, these patients are complicated. I don't know, they're staying in the hospitals too long. Physicians are not sure what to do with them. It wasn't any of that. It was like, okay, make it happen. You have the ability to change this course and make MUSC a destination center for this disease by integrating research, medicine, and education to really change what's possible for this disease. So thank you to all the patients. Thank you all for being here. Thank you for this wonderful day. Thank you, Dr. Rowland. I don't know if he's still around, for making this possible. It's really been an awesome opportunity. Thank you.